Great. Good afternoon, everyone. Welcome to the Dana-Farber Oncology Seminar Series. So I'm really delighted to introduce our speaker today, Dr. Lou Cantley. This is Ruiz and Shivik lecture, founded by the Shivik family. The late Ruiz and Herbert Shivik supported Dana-Farber as dedicated donors and volunteers for more than 40 years. Ruiz helped to establish the Friends of Dana-Farber Cancer Institute, an all-volunteer community that since 1976 has raised more than $47 million for research and care at Dana-Farber. In 2006, the Shiviks made a substantial gift to Dana-Farber to establish this annual seminar because they understood the importance of educating the next generation of oncologists. We are grateful to Ruiz and Herbert for everything they did for Dana-Farber over the years, and we are proud to hold this annual seminar, which bears their name. So now I introduce speaker Dr. Ruiz Cantley. So this is the man who needs no introduction. So I will be very brief. Lou received his PhD from Cornell, and then he did his postdoctoral research in Guidotti's lab at Harvard University. He became assistant professor, which is unusual, in biochemistry and molecular biology at Harvard University in 1978, and then associate professor in 1981. He was recruited to Tufts Medical School as a full professor in 1985. Then he came back to Harvard Medical School in 1992 as a professor of cell biology and cancer center director at the PI3 Medical Center. He was then recruited to Cornell Medical School in 2012 as the Mayo Cancer Center director there. So we are very lucky that Lou is coming back to Boston and then joining us at Dana-Farber and Harvard Medical School. Lou has made many significant scientific discoveries, including his discovery of PI3K in the mid-80s. His pioneering research has resulted in revolutionary treatments for cancer, diabetes, and autoimmune diseases. Lou has authored over 400 original papers and more than 50 book chapters and review articles. He received numerous awards and honors, including National Academy of Science, Medicine, and recently National Academy of Inventors. He received multiple AACR awards and prestigious awards like Breakthrough in Life Science Prize, Wolf Prize in Medicine, etc., etc. So my prediction is that Lou's next award is Nobel Prize. I think I'm not alone. Okay, Lou, so this I give to you, Lou's talk today is PI3K, insulin, and the cancer metabolism. Lou, it's yours.
Thank you, Jean. It's a pleasure to be back in Boston and get a chance to talk to my colleagues and also being next door to you and Tom. So it's a real pleasure. So I want to start by with my disclosure slide, and I should point out that I've started a number of companies over the years, some of them listed here: Petra Pharmaceuticals, Agios, and others. And in yellow, I just want to point out that one of the companies I started is is generating diets for use of cancer patients. And I will be talking about dietary intervention, so that's relevant for that conflict. And secondly, I will talk very briefly at the end about a mutant-specific PI3 kinase inhibitor that we developed at a company that I started called Petra that was recently bought by Loxo Lilly. So with that, I'll launch into my acknowledgements, and I'm in particular acknowledging in bold here Marcus Goncalves, Ben Hopkins, 2 postdocs in my laboratory, and in a paper that was a collaboration with Sid Mukherjee's lab, where we showed the first evidence that serum insulin levels are exacerbating the effectiveness of PI3 kinase inhibitors in cancer therapy in mouse models. And that led ultimately to clinical trials to test this idea that I'll talk about today. I also was fortunate to head a stand up to cancer team to test the alpha-specific PI3 kinase inhibitor in various clinical trials, including breast and endometrial cancers. In this paper with Ingrid Mayer, had the data coming out of that trial was, to me, very informative about the problem we would have in getting PI3 kinase inhibitors approved. So the outline of the presentation, I'll first talk about historical evidence for sugar enhancing incidence of cancer. And then I will focus on the mechanism, the biochemical mechanism by which sugar and insulin affects the ability of PI3 kinase inhibitors to treat cancers. I'm going to go back roughly 100 years to a scientist named Otto Warburg, whose photograph is shown on the right side here. And there was a book published just over a year ago by an author named Sam Apple about the life of Otto Warburg. And the title is Ravenous. And the book makes the point, which Otto Warburg really led the charge on, that consuming high rates of food, in particular glucose-rich foods, was causing cancers. And I'll show a little bit of the data on this, but this really was the first observation that cancers had different metabolism than other cells. And ultimately, this was called the Warburg effect, and it influenced thoughts about cancer when I was a graduate student back at Cornell many years ago. So in most mature tissues, adult tissues, when you consume glucose, most of the carbon atoms of glucose end up as pyruvate that enters the mitochondria and is oxidized to make ATP, oxidative phosphorylation. I should say that's what I worked on as a graduate student. And so this is how most normal adult tissues use glucose as an energy source. What Warburg observed was that if he took out a tumor from a rat and compared that to the tissue from which the tumor arose, that in the tumor, much of the glucose was going to lactate rather than to make ATP. But it was also being diverted into other intermediates, fatty acid synthesis, ribose, amino acids, glycerol. So rather than just using glucose to make ATP, glucose was being used for anabolic processes in the tumor. And this observation about 100 years ago that German scientists made was that there was this very large increase in glucose. And the reason that Otto Warburg looked at this is they were noticing that glucose consumption in Europe were going up dramatically, and that this was correlated with the increase in cancer rates. And that in contrast, if they looked at other populations, Eskimos, for example, in the Arctic, or certain tribes in Africa where Western diets had not yet appeared, the cancer rates were quite low. So they were beginning to think, even 100 years ago, that the change to a Western diet was influencing rates of cancer. So I will show you in the slide the data that actually existed and exists today in regard to obesity and sugar consumption. And you can see it's really quite dramatic. It wasn't until about 100 years ago, 120 years ago or so, I should say, that the sugar consumption rate went up dramatically. And that's shown in the circles. And we know the reason this happened was sugarcane became very cheap. It was produced in the Caribbean, where there's plenty of warm weather for the sugarcane to grow, and imported both to North America as well as to Europe, so that the availability of sugar to add to foods just went up dramatically. And hence you see the population more than almost 20 to 30-fold increase in consumption of glucose, as sugar. And following that, by about 10 or 20 years, was a dramatic increase in obesity in both North America and in this case, in Europe as well. So this correlation between sugar and obesity, we know, I think everybody believes that is true. But more importantly for this talk, there was a correlation also with mortality rates of cancer. So in the upper right, you see breast cancer mortality rates going up from 1920s to today, more than twofold. Increase in mortality rates from various types of cancers were increasing during this period of time in both Europe and the United States. So these are showing breast cancer rates, but endometrial cancer rates were going up even more dramatically. And perhaps the data that's most impressive, at least to me, is the data coming out of Taiwan. So prior to the 1980s in Taiwan, everyone ate a traditional Taiwanese diet that had been in place in China for hundreds of years. Plenty of rice, so plenty of carbohydrate, but virtually no sugar in the diet. Then with the entry of Western diets, the consumption of sugar started going up quite dramatically. And that correlates also with the dramatic increase in uterine cancers, endometrial cancers. And I'll focus quite a bit on endometrial cancers because they turn out to be the type of cancer that's most correlated with sugar consumption. But you can see that disease was almost non-existent in Taiwan and went up maybe almost tenfold over this period of time following the increase in sugar consumption at that time. And other types of cancers that aren't related to sugar consumption did not change. So I'll now get into the biochemical mechanism by which this correlation between sugar consumption, elevation of insulin, and the role of PI3 kinase in this whole process in causing cancers. So the first thing to think about then is what happens when you do consume sugar. So this little slide at the top shows the elevation in glucose levels you see in the bloodstream of someone who has a glucose monitor, and you follow what happens. And you can see that sure enough, you eat a meal that has some sugar in it, then you see an increase in glucose. And the glucose level in healthy individuals typically only goes up 10 or most 20% following a normal consumption of food. However, if you look at the bottom part of the slide in purple, the level of insulin is actually going up dramatically. And this, of course, is because as soon as the glucose level begins to climb even slightly, the pancreas begins to release insulin. And it will continue to release insulin until the glucose level comes back to normal again. So you get a long period of time with very high levels of insulin going up more than tenfold, while the glucose level is only going up a small amount. So this immediately begins to point to insulin as the more likely culprit for driving the growth of tumors than the elevation in glucose in the blood. So what does insulin do? So this slide is a very simple explanation for how insulin does almost everything that it does in liver, muscle, fat tissue, and other normal tissues as well, I should say. Insulin receptors are present in almost all tissues. So insulin binds to the receptor, which is a tyrosine kinase. It phosphorylates the adaptor proteins, IRS1, IRS2, IRS3, I just have one of them shown here. And the RS proteins, the sites that they get phosphorylated on, are optimal sequences for binding to the regulatory subunit P85 of PI3 kinase. The sequence is tyrosine, methionine, X-methionine, and then about 20 residues later, another tyrosine, methionine, X-methionine. So since methionine is a relatively rare amino acid, this context of 4 methionines within 23 residues or so, it rarely happens in any protein unless it's going to activate PI3 kinase. And I should say that this motif was first teased out in polyamido T in work that Tom Roberts was doing in collaboration with us back in the late 1980s. And it keeps showing up over and over again in every protein that activates PI3 kinase. So this brings PI3 kinase to the plasma membrane, and also the engagement of those two SH2 domains activates the enzyme. The catalytic subunit then, P110, is what produces the product of PI3 kinase, PI345P3, abbreviated here as PIP3. And that lipid, which is at the inner surface of the plasma membrane, then can engage proteins that have plextron homology domains that uniquely bind to that phosphorylated lipid. And the two shown here are AKT2, the serine threonine kinase, and its upstream activating protein kinase, phosphoinositide dependent kinase 1, or PDK1. They both have plextron homology domains that bind to the product of PI3 kinase, and that's how AKT gets activated. Now, I don't want to imply that everything that PI3 kinase does goes through AKT or PDK1. There are at least 20 other PIP3 binding proteins that are relevant for cell growth. But this particular signaling pathway begins to explain almost all of what insulin does. So if we look at some of the substrates of AKT, including GSK3, glycogen synthase kinase 3, which is involved in glycogen regulation, the FOXO transcription factor, which is involved in suppressing gluconeogenesis in liver and fat, TSC2, which is involved in the mTOR signaling pathway for protein synthesis, and AS160 in muscle plays a role in glucose trafficking to the plasma membrane. But I would say more important with regard to glucose uptake is phosphorylation of TXNIP, or thyrodoxine interacting protein, a substrate for AKT that we discovered about 10 years ago or so when Ning Wu was a postdoc in my laboratory. And phosphorylation of TXNIP by AKT leads to degradation of TXNIP. And the role of TXNIP in cells is to import glucose transporters to prevent them from bringing glucose across the plasma membrane. So the consequence of phosphorylating and degrading TXNIP is that glucose transporters stay on the plasma membrane for a much longer period of time. And I would argue that this is the primary reason why when you activate PI3 kinase, you get glucose influx into the cell. In other words, the Warburg effect is pretty much explained by AKT phosphorylation of TXNIP, although there are other events that AKT does that contribute to that as well. P10 is the phosphatase that takes off the 3-phosphate from PIP3 and returns the whole system back to normal again. And of course, P10 will result in prolonged activation of AKT and all the downstream responses that affect cell growth. So how often do we see mutations or amplifications of PI3 kinase in the gene encoding PI3 kinase, PIK3CA? I should just point out that there are multiple PI3 kinases that can make PIP3. Today, I'm going to be focusing entirely on PIK3CA, the one that produces the PI3 kinase 110 alpha that mediates insulin responses. So in green are mutations in PIK3CA, and in red are amplifications. And if you look at various cancers that have these events, you can see that uterine cancer, the one that I pointed out earlier, correlates most with obesity and insulin resistance. is the one that has the most mutations in PIK3CA. But other cancers, particularly women's cancer, cervical cancer, breast cancer, ovarian cancers, all have either amplifications or activating mutations in PIK3CA. But it's not unique to women's cancers. Colorectal cancers have PIK3CA mutations in both men and women at similar rates. So, but these are the cancers that we most pay attention to in regard to correlations with insulin activation, insulin elevation, and activation of PI3 kinase. If we add in loss of PTEN, which is shown in blue, single allele loss being light blue, double allele loss dark blue, now we see that a much wider section of cancers have at least 50% of the tumors with one of these events happening, either loss of PTEN through mutation or deletion, or activating mutations in PIK3CA or amplifications. So clearly, this pathway is getting highly mutated across a wide variety of solid tumors. So a few years ago, now I guess more than 10 years ago, I was fortunate enough to receive money from Stand Up to Cancer, along with Gordon Mills and Charles Sawyers, to start up a team to explore PI3 kinases in women's cancers, and particularly breast cancer. And several of you in the audience, I hope some of you are in the audience, have been my colleagues on that team for many years. Eric Weiner, previously, who's now left, but played a major role in leading the clinical studies in this on this Stand Up to Cancer team. This is one of the first patients to go onto our trial, and this is the time when alpelisib, the PI3 kinase inhibitor that ultimately was approved, was first going into phase 1 studies. And this woman had an FDG PET. She had breast cancer and she had mets to the liver. You can see these black spots in the liver indicating glucose, dramatic increase in glucose uptake into those mets. And she then went on alpelisib, the PI3 kinase inhibitor, also called BYL719, now called PICRE, approved drug. And you can see that the liver mets went away acutely. Although this did not cure this woman, the tumors ultimately came back and she progressed. So, but this was evidence to me that this drug, you could achieve a dose of this drug that hit PI3 kinase and turned off glucose uptake into the tumor. And therefore, I was encouraged that we should work with Novartis. Our team should work with Novartis in designing and executing the phase 1 studies. Now, I should say that for the phase 1 studies, we decided that we were aware that insulin would activate the mutant PI3 kinase even better than the wild type, and therefore insisted that the patients not receive insulin injections during their phase 1 study. And the reason being that if you have insulin elevation as a consequence of glucose elevation, then that insulin is going to activate PI3 kinase in the tumor even more readily than it does in normal tissue. So this axis of consuming glucose, pancreas elevating insulin levels, and insulin then activating through the insulin receptor PI3 kinase in the tumor, in the normal tissue, which helps bring glucose back to homeostasis. The same cycle can happen in the tumor if it has insulin receptor, but this is going to drive glucose into the tumor, and that's why the FDG PET signal works. This elevation in insulin is causing PI3 kinase to drive glucose uptake into the tumor. Now, if the patient happens to be insulin resistant, then the pancreas has to churn out a lot more insulin in order to bring glucose back to homeostasis, because the liver or muscle are not responding properly to insulin. And that elevation in insulin is going to even further drive the growth of the tumor. And I would argue that this is why there's been strong correlation between obesity, insulin resistance, and certain types of cancers, including endometrial cancer, It's a consequence of this elevated insulin when the patient is insulin resistant. And if the patient is insulin resistant and is also on a high glycemic diet, in other words, consuming a lot of pasta and sugars, sugary drinks in particular, this is going to further exacerbate the elevation of insulin and drive tumor growth. So if we look then historically at patients who are type 2 diabetic, and also in all type 2 diabetics will have gone through a period of time of insulin resistance before they progress to type 2 diabetes. So type 2 diabetes is defined by the fact that you have to give some kind of treatment to keep the insulin, to keep the glucose levels down. And that treatment can be insulin injections, but it can also be metformin or sodium glucose cotransporter inhibitors, which allow the glucose to be cleared into the urine. In any event, the subset of patients who ultimately develop type 2 diabetes after having gone through a period of insulin resistance, you can see are at much higher risk for developing endometrial cancers. And these are a number of independent studies done in different places, Europe, US, in different settings, different age groups, et cetera. And in every one of these studies, you can see a higher risk of endometrial cancer, up to twofold or more for patients who had gone through a period of insulin resistance. I should also point out that although we don't typically look for this in patients, if you actually go through the CBio portal database and ask how often do you see insulin receptor amplified or IRIS1 or IRIS2 amplified, it's actually a significant number of patients who have this event going on, indicating that producing more insulin receptors or IRIS1 or IRIS2 in the tumor itself must be contributing to tumor growth. And sure enough, if you then look at the overall survival, the patients who have these amplifications have a far worse outcome, shorter survival rate. Okay, so what can we do about this? How can we manage glucose levels and still without raising insulin levels? The endocrinologists don't care about raising insulin levels. They very often give patients insulin to keep their glucose in control. But I worry about it because of the previous slides that insulin will drive PI3 kinase, particularly mutant PI3 kinase. So these are some studies we did in mice that they look identical to what we saw in the early clinical trials 10 years ago when patients went on the various PI3 kinase inhibitors that were available at that time. The patients all had elevation in glucose levels, and they tended to peak about an hour or two after taking the drug. So we checked in mice to see whether what we saw in human patients could be replicated in mice, and sure enough, we see the same thing in mice. You give any one of these PI3 kinase inhibitors that had gone into phase 1 studies, and glucose levels went up more than twofold. So what we were really interested in knowing, though, is what's happening to the insulin level in both the patients and also in the mouse patients. And I'll point out that at about 2 hours or so, you begin to see the glucose levels starting to drop. So why is the glucose dropping? I would argue it's because insulin levels have gone up high enough to override the inhibition of PI3 kinase. So keep in mind that in normal tissues in your body, if you give a PI3 kinase inhibitor that inhibits both wild type and mutant, you're going to shut off the ability of all these tissues to respond to insulin. So you can't suppress gluconeogenesis in the liver. You cannot take up glucose into the muscle, and as a result, glucose levels are going to go up and insulin levels are also going to go up. And sure enough, if we look at insulin, you see this dramatic increase in insulin in response to giving any one of these PI3 kinase inhibitors. BYL719, I should point out, is the alpelisib drug that's now approved with the name Piqray. And you can see it causes a quite dramatic increase in insulin in the mice, and we see the same thing in patients in the phase 1 clinical trials. So why am I worried about this insulin? Well, if you take an endometrial tumor and grow it ex vivo, these are organoids, PIK3CA mutant endometrial cancer patients at Weill Cornell. We grew up the organoids, and then we treated them with the PI3 kinase inhibitor, in this case, the pan PI3 kinase inhibitor, BKM120. And you can see that at about 1 or 2 micromolar, we see an acute death of these endometrial tumors. As long as we didn't have any insulin in the media, this is of course done ex vivo. However, if we add the level of insulin you would see in a patient at about 2 hours after giving this same drug, that amount of insulin, roughly 10 nanograms per mil that you would see in the serum of that patient, allows that organoid to survive in spite of the presence of the PI3 kinase inhibitor. So in other words, the insulin receptor in that endometrial tumor is getting so active that the drug cannot completely turn off PI3 kinase. And we can show that by a variety of means here. If you look at phospho-AKT, you can see that when you give insulin, it will activate PI3 kinase. If you add the inhibitor, instead of giving insulin, you turn off the basal PI3 kinase phosphorylation of AKT. However, if you give the amount of insulin you would see at 2 hours after the patient had taken that same drug, now the phospho-AKT is actually higher with the insulin level elevation in the continued presence of the drug than it would have been had you given no drug at all. So you're actually activating AKT by giving a PI3 kinase inhibitor as a consequence of this dramatic overshoot in insulin that's seen in the patients, in both human patients and mouse patients. And this results in downstream signaling to phospho-S6 also being up, so the whole pathway is turned up in these tumors. So that's the problem. So the question is, how can we bring the glucose level back down? Because these elevated levels of glucose are quite harmful. How can we bring that glucose back down without elevating insulin? So obviously, we don't want to give the patients insulin for all the reasons I've just told you about in the last 10 slides. So what are other choices? Well, at the time we started our trials, metformin was the only really good drug other than insulin for treating patients with type 2 diabetes or insulin resistance. It's a very safe drug. It goes to the mitochondria and suppresses mitochondrial function, and this is ultimately how it works. But there are other drugs available now that weren't available 10 years ago, including a sodium glucose cotransporter inhibitor that I mentioned briefly earlier. So these inhibitors go to the kidney and turn off the transporters that use the sodium gradient between the blood and the kidney urine to drive glucose back up into the serum. So this is how your body avoids all of your glucose ending up in the urine. If you inhibit these transporters, then much of the glucose will end up in the serum. So that's now an option that wasn't available 10 years ago. There are now 3 different drugs for SGLT2 inhibitors that are approved for use in humans. PPAR gamma effectors have some toxicity side effects. We avoided using those. But the other intervention that we consider were what we call ketogenic diets, not because of producing ketones, but because they have such a low amount of carbohydrate in them that they don't raise insulin levels as much. And therefore, the patient is living more on energy provided by the ketones than from glucose. So we explored all these possibilities in our mouse models, and you can see that sure enough, the sodium glucose cotransporter inhibitor is very dramatically lowering blood glucose levels. On the left side and on the right side, you can see it also lowers insulin levels as judged by C-peptide levels in the serum of the mice being treated with this inhibitor. So, and of course, at time 0 is when the PI3 kinase inhibitor is added. It causes this elevation in glucose and elevation in insulin. Metformin, which we used in our phase 1 clinical trials 10 years ago, also can somewhat blunt the elevation in glucose, but you can see it's not nearly as effective as the sodium glucose cotransporter inhibitor, or as effective as the ketogenic diet, which has low carbohydrate. And you can see, again, by C-peptide levels, that metformin does not lower insulin nearly as much as the sodium glucose cotransporter inhibitor. And the ketogenic diet is actually the best way to keep insulin levels low. So this is, again, is an example in an endometrial tumor that has PTEN deleted and a PIK3CA mutation. These turn out to happen simultaneously in many cases. And if you look at this tumor growing in a mouse and just look at normal conditions with the tumor growing in the mouse and stain for phosphorylated insulin receptor, antibodies against the phosphorylated receptor, you can see this brown stain that there's quite a bit of insulin receptor being phosphorylated, indicating that the ambient insulin level in that circulating blood into that tumor was enough to keep the insulin receptor somewhat active. If you switch this mouse to the ketogenic diet, which has only about 5% carbohydrate, then you see only gray, very little brown stain, indicating you've dropped the insulin level and you've dropped the amount of phosphorylation of insulin in the tumor. If you give, however, the PI3 kinase inhibitor BKM120, you dramatically raise insulin levels because the glucose level goes up so much. And the consequence is you have massive activation of the insulin receptor in the tumor. But the ketogenic diet will dramatically drop this by preventing the PI3 kinase inhibitor from elevating glucose and insulin. And as a consequence, you now have turned off the insulin receptor quite dramatically in the tumor. So if you look downstream at phospho-S6, indicating that you've activated the PI3 kinase inhibitor, also activating mTOR downstream of the PI3 kinase pathway, you now see that, again, adding BKM120, you actually increase phosphorylation of S6, which is why this drug doesn't work so well in the tumor. Metformin helps bring that down some, but the sodium glucose cotransporter inhibitor or the ketogenic diet dramatically reduce the phosphorylation of the insulin receptor, or of S6 rather. So you're bringing down phosphorylation of insulin receptor, and you're bringing down the downstream pathway to phospho-S6 is also getting shut down. So the question then is, do these interventions, either the sodium glucose cotransporter inhibitor or the ketogenic diet, improve responses to PI3 kinase inhibitors? And this is the result, and it's not subtle. So again, these are mice. These are tumors growing in mice. This is actually a PIK3CA mutant triple negative breast cancer allograft. And you can see that all these various colors here have very little difference in the rate of growth of the tumor, including adding the approved drug, alpelisib, also called PICRAE, BYL719 it was originally called. That's the blue line, and you can see the blue line is somewhat shifted from the control green line. You get an extra 5 days or so of lag in tumor growth, but nothing to write home about with this drug as a single agent in this tumor. However, if you put that mouse on a ketogenic diet for 2 weeks before you start the drug treatment, now the tumor, rather than growing, actually shrinks and essentially disappears. So by keeping this feedback effect of insulin activating PI3 kinase in the tumor through insulin receptor, you can now make these drugs that have only some retardation of growth of tumors into actual home runs, at least in mouse models. So the question is, can we make this happen in human studies? So this drug, alpelisib, as I pointed out, is already approved PICRAE in combination with fulvestrant for treating ER-positive PIK3CA mutant breast cancers. And the truth is, though, this drug compared to fulvestrant alone only gives about a 6-month progression-free survival effect on these women who have PIK3CA mutations. So that's good. It's enough to get it approved, but can we do better? Can we actually shrink those tumors and cause many years of progression-free survival rather than 6 months? And so that's the study that we would now like to do to see whether what has played out so beautifully in the mouse studies will also work in human studies. And I should add, the evidence that this has nothing to do with ketones, it's really the insulin, is that if you inject insulin IP into the mouse to get the insulin level back to where it would have been had you not been on a ketogenic diet, the tumors go right back to growing at the same rate. So it's all about the insulin driving the tumor growth, not the ability to produce ketones on a ketogenic diet. So this has led us into a number of clinical trials, and I should say that Marcus Goncalves, as I mentioned earlier, has played a major, he's an endocrinologist, an MD, PhD endocrinologist. He's trained as a postdoc in my laboratory. He's now an assistant professor at Weill Cornell. And he really designed these trials that I'm describing here. One is with Bayer's alpha delta inhibitor. It's already approved because of hitting delta for AML. And is now being explored in endometrial cancer with a trial that's being enrolled at Columbia, Weill Cornell, and Sloan Kettering that was designed by Marcus Goncalves. And then an additional trial that we're just starting to enroll patients in. I should say the endometrial trial has already enrolled more than 18 patients, but an additional trial in breast cancer is now open at Weill Cornell, Sloan Kettering, and Ohio State that will utilize the Novartis PI3 kinase inhibitor alpelisib for patients who are ER positive, HER2 negative, and PIK3CA mutant. And this trial has now enrolled 3 patients, slowed down a little bit by COVID, but hopefully we'll get accelerated enrollment over the next few weeks to months. So with that, then I do want to show a couple of slides of the clinical collaborators in this work. Peter Martin, who's worked in AML, Jennifer Amengal and Vicky Macker in endometrial cancer, played a major role in helping with Marcus Goncalves to design and is now enrolling patients in these trials. These are in the endometrial and lymphoma trials. And in the breast cancer trials, Eleni Andropoulou from Weill Cornell, Neil Inyengar from Memorial Sloan Kettering, and Miriam Lusberg from Ohio State are the site leaders for the breast cancer trial. Overall lead being Neil Inyengar. And this trial, as I say, has, the last time I spoke with them a week or so ago, had 3 patients enrolled and several others being considered for the trial. So we're optimistic that we'll see some very positive results in these studies, and hope to really have a change in how patients can respond to these drugs by dietary intervention. I then just want to end with another obvious question to ask, and that is, if you had a drug that only hit the mutant PI3 kinase and didn't hit the wild type, then you wouldn't get this insulin overshoot. It's because you're turning off PI3 kinase in liver and muscle and fat cells that the glucose level and ultimately the insulin levels go up in the patient. If the drug only hit mutant PI3 kinase, then you would not see this rebound of glucose uptake elevation and insulin elevation. And the drug, as a consequence, should be much more effective. You wouldn't need a dietary intervention or a sodium glucose cotransporter inhibitor in order to get an effective response. So based on that concept, I started a company called Petra Pharmaceuticals in New York City about 5 or 6 years ago in collaboration with Nat Gray. And we spent a lot of time looking at mutant PI3 kinase to see whether there might be a way to develop a drug that hits only the mutant enzyme and not the wild type. And in the end, came up with a small molecule that specifically hits the H1047 mutant of PI3 kinase. And this accounts for about 40% of the mutations that you see in PIK3CA are at this site. So this drug would only be beneficial for patients that have this specific mutation, in that it does not affect either wild type PI3 kinase or the other mutations like the 545 mutation. So we moved forward with this compound, and it looked incredibly effective in studies in mice with tumors that had 1047 mutations. There was no elevation in glucose or insulin at all in the bloodstream, and the tumors completely disappeared without having to do dietary intervention. Petra got purchased by Loxo Lilly a little over a year ago. And they've now, this drug is, or this molecule, I should say, is now in human clinical trials. And so hopefully this will be a benefit for the patients who have this particular mutation. So with that, then I will finish again with the acknowledgements, and glad to take questions.
Great. Thank you, Lou. This is amazing. And so the audience, that if you have questions, please post in the Q&A. So Lou, I'm going to read a few questions already there. Can you hear me well?
Yeah? Yes, I can hear you.
Okay. Yeah. Okay. The first question: do you think that high concordance of endometrial cancer with obesity, high insulin states, has something to do with the innate biology of the endometrium, e.g., repeated cycle of hypertrophy, deciduation?
Yeah, I think this is true. Obviously, with the menstrual cycle, you have growth and depletion of those cells every month. And there's certainly pretty high levels of insulin receptor in the endometrium. And so that being able to grow that tissue so dramatically every month clearly depends on activating PI3 kinase. So it's set up to be highly responsive throughout at least the period of being able to have children. So up through the age of 50 or 60 or so. And this continuous cycle every month is of high proliferation is, I think sets up the endometrium to be likely to accumulate some mutation. And once you get to mutation, because of the high insulin receptors, the tumors take off growing. So I think the correlation with obesity is really due to elevated insulin levels.
Okay, that's great. So I just switched the order because that question from Alan Huang, and he has another question. Can you achieve equivalent synergy with SGLT2 inhibitors plus alpelisib compared to ketogenic diet plus alpelisib?
Yes, pretty much the same. So you could argue, and in clinic, I should say that the clinical trial that I described actually has 3 arms to it. One arm is just standard of care with fulvestrant plus the alpelisib. That's the control group. The second arm has the ketogenic diet. I should call it insulin lowering diet. And then the third arm is the sodium glucose cotransporter inhibitor. No dietary intervention. Although the patients, for all 3 arms, the patients are advised not to take sugary drinks with their pills and to, avoid sugar in general. But the strict diets are refined to only one arm of the trial, and sodium glucose cotransporter to another arm of the trial. So we think the trial is well designed with sufficient number of patients calculated that we should be able to reach a conclusion that would allow us to have approval for sodium glucose cotransporter inhibitor or a ketogenic diet being required or even reimbursed for patients who are taking alpelisib.
Great. Okay, next, a naive question. Does the drug inhibit glucose uptake will affect the readout of PET scan? A false negative result?
Yes, that's right. In fact, I would say that PET scan was a false negative result. That the tumor was still there. It wasn't taking up glucose because the PI3 kinase inhibitor was blocking the glucose uptake. So yes, that is a false negative. Now, the truth is the drug was also prolonged, as I say, the drug gives about a 6-month progression-free survival. So it's not that it's not doing anything on a normal diet. So it's hard to, in looking at the FDG PET, how much of this is due to the acute turnoff of glucose influx and how much is due to the tumor actually starting to die. And you can't tell from that assay alone.
Okay, next question is from John. Thanks for the great talk, Professor Kentley. I am curious about your current study that couples of the ketogenic dieting with alpelisib. Typically, people who are on keto diets for weight loss won't see effect for at least a month. How long do you have the mice or participants on the keto diet until you administer alpelisib? Do you see a linear correlation or some sort of correlation of time on keto diet and the effect from alpelisib?
Yeah, I think I mentioned when I showed that slide that the mice were on, I can't remember whether it was 2 weeks or 1 week, but they were on at least a week of the ketogenic diet before they went on the drug. And the reason is you need about that period of time to go to ketosis, but we don't think it has anything to do with ketones. We just wanted to deplete the glycogen stores.
Right. Okay. Next, thanks for a beautiful talk. In thinking of ways to lower insulin glucose levels to boost PI3K inhibitor therapy, were insulin receptor inhibitors ever considered? Any reason why this didn't or couldn't work?
Yeah, so insulin receptor inhibitors have been developed and gone into clinical trials. Inhibitors that hit both insulin receptor and IGF-1 receptor, as well as IGF-1 receptor alone. So there's a reason for all of those. IGF-1 receptor, well, first of all, many tissues have covalent heterodimers of insulin receptor bound to IGF-1 receptor. So there's a disulfide bond on the extracellular domain that covalently links either insulin receptor homodimers or IGF-1 receptor homodimers or insulin receptor IGF-1 receptor heterodimers. It all depends on the relative abundance of insulin receptor versus IGF-1 receptor on the cell surface as to which you get. But in most cases, you've got a whole spectrum of pure insulin receptor homodimers, pure IGF-1 receptor homodimers, and heterodimers of insulin IGF-1 receptor. So there's been a lot of, more focus on IGF-1 receptor inhibitors than insulin receptor inhibitors. But there have been drugs that hit both, gone into clinical trials, and they failed. And they failed because the insulin levels go up so high if you turn off the insulin receptor that ultimately the insulin can override whatever inhibitor you have. And if it doesn't, the patients become so hyperglycemic that the endocrinologists force them to go off the study. If you're hyperglycemic for a long period of time, you lose circulation in your legs and feet and retina. And so you have to have amputations of all of your limbs and you lose your eyesight. And the kidneys also quit working. So you don't want a prolonged period of high levels of glucose in your bloodstream. So that's the real problem with using inhibitors of insulin receptor.
Okay. Next is from Lee. Is insulin has similar effect on prostate cancer. If a tumor-bearing mouse has insulin resistant, does the immune environment change?
Say that again.
Okay,
The prostate
so yeah, first is part is prostate cancer. If insulin has a similar effect on prostate cancer, because you talk about breast and endometrial cancer, where this has similar in the prostate cancer.
Yeah, I would say absolutely for the following reason, although we've not done as much studies on, although we've generated mice with, with P10 deleted prostate tumor and done some studies on those, not as much with in recent years where we started doing the interventions with dietary studies. So I can't speak to the mouse models as much there, but I can tell you that for prostate cancer patients, actually, I learned this from epidemiologists at Dana-Farber, actually, that for prostate cancer patients who go on anti-androgen therapy and typically gain weight, if you lose your androgen receptor signal, then almost every man starts to put on fat. And that gaining of weight, if they significantly gain weight, they have a much shorter survival period after androgen receptor ablation than those who manage to keep their weight under control. And that's just an epidemiological observation. I would argue that what that means is that if you start gaining weight, your insulin levels are going to go up as you become insulin resistant. And if you can keep a dietary intervention or exercise that prevents that elevation, that gain in weight, then you'll maintain insulin sensitivity. Insulin levels will stay low, and that will reduce the rate of growth of that tumor. So that's, again, very speculative because I don't have mouse studies to back it up, but I think that's what's going on.
Yeah, so the other half of the question is, if a tumor-bearing mouse has insulin resistance, does the immune environment change?
Good. Actually, let me finish off there was one other thing I was going to say about the prostate cancer, and that is that P10 loss is extremely frequent in late stage disease. So in early stage prostate cancer, you don't see P10 loss, but P10 loss is quite elevated. It's the most significant thing that you see in late stage prostate cancer. So I think that's why you're getting such an insulin response, because you can't turn it off. You can't turn off PI3K with P10 loss. So the second question was about
immune microenvironment, if it has an insulin resistance.
Yeah, immune microenvironment, that's really an interesting question. We've thought a lot about whether the PI3 kinase inhibitors themselves are having an effect in the immune microenvironment. So the advantage of alpelisib is that it doesn't hit the delta form of PI3 kinase. So the immune cells, the PI3 kinase that's relevant is PI3 kinase delta in B cells and T cells, and PI3 kinase gamma in monocytes and general macrophages. And so the alpha specific inhibitors don't have, although there's some alpha, PI3 kinase alpha in B cells, and in fact, it's when the delta inhibitors fail, in some cases it's because the alpha, PI3 kinase alpha is upregulated or mutated even. But it's not the dominant PI3 kinase in immune cells. So you're not, probably not having a big effect on blocking T or B cell mediation in the tumor microenvironment. The macrophage, again, not a large effect. So I don't think, but the question really was insulin. Is insulin regulating macrophages or T or B cells? I've looked into that a bit. There are some insulin receptors in immune cells, but they're not nearly the level you see in solid tumors or solid tissues like endometrium or breast.
Okay, so next is also sort of immune related. Thank you for the talk. Insulin certainly play a role, but how large of an effect do you think that inflammatory cytokines and immune suppressive effects from the obesity have on cancer development?
Well, the question is what causes inflammation. So this is maybe somewhat orthogonal to the question, but my brother, who's a nephrologist at Yale and does research on kidney repair, has published quite a bit on the role of macrophages, monocytes, on repair processes. So I always think that tumors, there's a century-old saying that cancer is the wound that will not heal, that the same processes that drive wound healing are involved in tumor growth as well, at least in the case of solid tumors. And I think that's absolutely true. What he sees, if he induces an injury in the kidney in a mouse model, and then allows blood flow to come back and watches the whole repair process, the first thing that happens is the macrophage comes in. And the macrophage, by directly contacting the adjacent normal tissue in the kidney, will drive it to become a stem-like cell and start dividing. So that's the first thing the macrophage does. And he has evidence. You physically have to get contact between the macrophage and the adjacent normal tissue in order for that tissue to start growing again. And then secondly, so this is not stem cells getting reactivated. These are differentiated cells becoming de-differentiated and start dividing again. Then the second thing that happens is the macrophage forces the T cells and B cells out of the wound area in order to avoid an autoimmune attack of the injured tissue. So if you think about that tissue repair process I just pointed out, and you now think about a cancer that the macrophage looks like an injured tissue, it's going to do the same thing. It's going to keep the T cells and B cells from attacking it. And at the same time, it's going to help stimulate its growth. And I think there are lots of studies that numerous laboratories have done to show that this is actually what does happen in tumors as they initiate. In liver models, I've seen studies on this. And even in metastasis, it's the macrophage that helps keep that metastatic cell alive when it's vulnerable and help it get out of the bloodstream and into the tissue. So the macrophage is our enemy when it comes to cancer. It's our friend if it comes to wound healing.
Absolutely. Thank you so much, Lou. I think that there are a couple of questions left. I think time is up. Claudia will forward these to you. And I think that we thank you again, Lou, for the really amazing talk, and thank everybody.
Thank you. It's a real pleasure
to
get to talk to everyone, to be back in town again. And I'm looking forward to meeting with all of you.
Excellent. Great. Thank you.